Okay, welcome back for lesson 3 in our unit on natural selection. Today we're going to discuss population genetics and specifically Hardy-Weinberg equation and Hardy-Weinberg principle. Just a quick introduction. Population genetics is a field of study which seeks to better understand exactly how natural selection brings about evolutionary change and also uh, seeks to better understand other factors besides natural selection which may contribute to evolutionary change within a population. We know natural selection is the driving force behind evolution. We know that because of the work of Charles Darwin, but uh, there are things that we've learned since the time of Darwin's publication. We know that it's a little more complicated than even he realized, and there certainly are some other factors besides natural selection that play a role in evolutionary change. Okay, so some terminology. It's always good to start out with some terms that you need to know. So population genetics, I mentioned that term before. It's the study of genetic changes in populations. And the concept of population genetics is one of many ideas that has become known as the modern synthesis. Sometimes it's referred to as neo-Darwinism, and it's a comprehensive theory of evolution that was formed sometime around the 1940s. It takes Darwin's ideas from his 1859 publication and kind of puts it together with all of the genetics knowledge that we've gained since that time for a better understanding of how these underlying factors work along with natural selection to create genetic change over time within populations. Okay, as we move down the road in population genetics, you'll hear me use the term gene pool a lot. You've heard that term before, I'm sure. The gene pool is the sum of all the genes within a population at any one time, and very often it's used in a context where you're really only interested in the genes within that population for a particular trait. So I'll give you some examples in just a few minutes for how we might use that term. Another one that gets tossed around a lot is allele frequency or allelic. Sometimes instead of an E on the end, you'll put IC, allelic frequency. It's the proportion of a particular allele within the gene pool compared with all the alleles for a particular gene. So let's say we're talking about pea plants, and we're talking about tall and dwarf pea plants, so big T for tall and little t for dwarf. And so let's say you've got hundreds of pea plants in a garden. The gene pool would be like, out of all the genes for height, what proportion of them are big T? And what proportion are little t? And again, that'll make a little more sense when we start looking at examples. We're going to talk about the Hardy-Weinberg equation, which is math. It's the mathematical way that we can calculate and predict allele frequencies and also genotype frequencies within a population from one generation to the next, assuming that evolution is not occurring within that population. And then related to that mathematical concept of Hardy-Weinberg equation is an idea called the Hardy-Weinberg principle. And that is a theorem which seeks to identify which forces may be causing allele frequencies to change, certain alleles becoming more common, other alleles becoming maybe less common from one generation to the next within a population that is evolving. In other words, if evolution is causing genetic change, we want to know, is that just due to natural selection, or could it also be due to some other factors, things that Charles Darwin didn't even know about, that we have learned more recently. And so it's a very interesting topic. It has a little something for everybody. If you're a math-minded person, you're going to like a lot of what we do in this lesson because we're going to do a lot of math problems. But there's also some good sort of critical thinking, solving the mystery of what's causing the genetic change within the population. So I've always enjoyed this aspect of evolution. Now we've defined evolution before. So within the context of population genetics, we can tweak that definition just a little bit. We can refine it a little bit. And instead of just saying evolution is change in a population over time, now we can be more specific. We can say, well, evolution is when certain alleles are becoming more common or less common within a gene pool from one generation to the next. Or another way to say that, evolution is change in allele frequencies within a population through time. Now, as we've discussed before, natural selection acts upon individuals, right? Natural selection determines which individuals live longer, reproduce more successfully. Natural selection kind of calls out the organisms, the individuals that are not as adapted to live, survive, and reproduce. It's the individual's characteristics that affect its chances for survival and reproduction. But the impact, the result of natural selection is manifested not on those individuals, but on the population as a whole. So we've talked about that before. Individuals do not evolve. It's the population that evolves, or the whole species that evolves. Okay, so I mentioned the idea of gene pool and allele frequency. So now let's have like a practical example. So here's a little garden plot, and somebody has planted some pea plants. And as you know, big T for tall and little t for dwarf. And so, the tall plants are like 6 feet tall when they reach full adulthood, and the dwarf plants are only a couple of feet tall. And so in this particular garden, it looks like there's 2 dwarf plants, one here and one here, and the rest are tall. And some of the tall plants are homozygous tall, and some are heterozygous. So in this population of 10 plants, of course, each plant has 2 genes for height. And so that's a gene pool of 20, okay? And so we could calculate the allele frequencies. You see this little p right here? Now this is confusing because, we're talking about pea plants, and I don't want you to think, p for purple or little p for white. We're only thinking about the height of the plant right now. So forget about purple and white. Lowercase p in the Hardy-Weinberg equation is just a generic letter that represents the frequency of the dominant allele. In other words, in this gene pool, how common is the dominant allele? Well, we can count them up. How many big T's are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So out of 20 genes in the gene pool, 12 of them are big T. 12 divided by 20 is 0.6. In other words, 60% of my gene pool is big T. Of course, the other 40% is going to be little t. Lowercase q in the Hardy-Weinberg equation stands for the frequency of the recessive allele. So these two numbers are always going to add up to 1, 0.6 plus 0.4. But just to double check, let's count the little t's. 1, 2, 3, 4, 5, 6, 7, 8. Sure enough, 8 out of 20 are little t. That's the frequency of the recessive allele, and it's 0.4. Okay, let's look at another example. Here we have a population of some kind of flowering plant, and it looks like big R dominant allele codes for red flowers, and recessive allele, little r, codes for white. And so let's see, we got a population of 500 plants. So that means our total number of genes is going to be 1,000. You see that 1,000 down here? Because think about it, if there's 500 plants, every plant has 2 genes. 1, 2, 1, 2, 1, 2. So 500 plants, they each have 2 genes. That's 1,000 genes for flower color in the gene pool. So if we know some statistics, like if we know that 320 of the plants are homozygous red and 160 of the plants are heterozygous red and 20 of them are white homozygous little r, we can calculate not only allele frequencies, but we can calculate what are called genotype frequencies. So anyway, if I want to know what percent, or what, I shouldn't say percent because we're not actually going to express this as a percent. We're going to just express it as a decimal. So it's called a frequency, not a percent. So sometimes I slip up though, and I'll refer to it as a percent. The frequency of the homozygous dominant genotype would be 320 plants divided by a total of 500 plants, because if you add all these together, it's 500 plants. 320 divided by 500 is 0.64, okay? Or if you wanted it to be a percent, of course, that would be 64% of the population. I can calculate the frequency of heterozygous individuals, which would be, in this case, 160 divided by 500, which is 0.32. And I could also calculate the frequency of homozygous recessive 20 plants divided by 500 is a very small frequency, 0.04. If you add those 3 numbers together, notice 0.64 plus 0.32 plus 0.04, it adds up to 1. This is the premise behind this equation, this wonderful mathematical equation called the Hardy-Weinberg equation. And what this basically says, in any population that is not currently evolving, if you add up p squared, which is the frequency of homozygous dominant individuals, plus 2pq, which is the frequency of heterozygous individuals, plus q squared, the frequency of homozygous recessives, that's everybody. So that's got to add up to 1. And you can see it absolutely does, 0.64, 0.32, and 0.04. And if this population is not experiencing evolutionary change right now, if there's no natural selection, there's no forces causing this population to evolve, then when these plants all start interbreeding and pollinating each other and producing offspring, the next generation should have these same numbers, these same genotype frequencies. Again, that's assuming that evolution is not taking place. Normally, populations do experience evolutionary change, but let's just assume, hypothetically, that there's no evolution taking place. These numbers should remain stable 1 generation to the next. Now, I mentioned the idea of P and Q. Remember, P is the frequency of the dominant allele, and you can see they've calculated that to be 0.8. Q is the frequency of the recessive allele, which they calculated to be 0.2. Let's refresh our memory how they did that. So if I want to know the value of P, the frequency of the dominant allele, well, let's think about it. There's 320 of these homozygous dominant plants. They each have 2 dominant alleles, so that's 640 copies of big R. There's 160 heterozygous plants. They each have 1 copy of big R. So that's 160 big R's plus 640 big R's. That means in the whole gene pool, there's 800 copies of big R. Look at that again. 320 plants like this, and they each have 2 copies of big R. So that's 640 big R's. And then the heterozygous plants have 1 copy. There's 160 of them. So that adds up to 800. 800 divided by 1,000 genes in the gene pool is 0.8. Okay? If I want to calculate the value of Q, that's going to be the number of little r's in the gene pool divided by the total number of genes. So 160 heterozygous plants, they each have one little r right here. And then there's 20 of these guys, and they each have two little r's. So that's 40 more. 160 plus 40 is 200. And there's 1,000 genes in the gene pool. So 200 divided by 1,000 is 0.2. P plus Q must always equal 1. Those numbers will remain stable from one generation to the next, assuming that evolution is not occurring. Remember, P plus Q always equals 1. P squared plus 2PQ plus Q squared also always adds up to 1. And let's remember what that means. P squared, whoops, it disappeared on me. P squared is the frequency of homozygous dominant individuals. Out of all the flowers in that population, P squared is how many of those plants are not just dominant, not just red flowers, but homozygous dominant, big R, big R. Okay? 2PQ is the frequency of heterozygous individuals, and Q squared is the frequency of homozygous recessive individuals. If you add those up, it has to add up to 1. Okay, and so where are we headed with this? Well, you need to be able to calculate allele frequencies in basic Hardy-Weinberg problems. You're going to see some of these on the AP exam. They're kind of fun when you get the hang of them, and some of them are just dirt easy, and some of them are a little more challenging. But here's the thing. You don't have to memorize this equation. I mean, it's not a hard one to memorize, so it's probably easier if you do just go ahead and commit it to memory. But they're gonna give you this equation on the AP exam, okay? And every Hardy-Weinberg problem like this, they have to give you something. They're either going to give you Q squared, and you would then take the square root of that to find Q, and then everything else will be based off of that number. Or they might just give you Q. Well, see, if they give you the value of Q, it's pretty easy to figure out what P is, right? If they tell you Q is 0.5, then P also has to be 0.5 because these two have to add up to 1. Or if they tell you what Q squared is in the problem and you want to find Q, just take the square root of it. You can use a calculator. You're going to need a calculator for these. So let me show you how easy this can be. Assume that 9% of people are unable to taste PTC. That's little t, homozygous recessive if they cannot taste PTC. What are the allele frequencies for big T and little t? All right, and we also want to figure out what percent of the population are homozygous tasters. Oh, and we want to also know what percent of the population are heterozygous tasters. So the first thing to realize is if you have a person that cannot taste PTC, they have to be homozygous recessive. So they're giving us the frequency of homozygous recessive individuals in this population. They're telling us Q squared is 0.09. Not 0.9, because that would be 90%. It's 9%, which is 0.09. Okay? Now, can you find Q? Just put 0.09 in your calculator and hit the square root button. And look at what you get. 0.3. Now, 1 minus that is going to give me P. So P is 0.7. And I hate that this is animating this way. It's kind of irritating. I'll see if I can fix it before we do the next problem. But it says, what are the allele frequencies for big T and little t? The frequency of big T is P, frequency of the dominant allele. We already calculated that right here. Frequency of the recessive allele is Q. We already calculated that right here. It's 0.3. What percent of the population are homozygous tasters? That's p squared. So if you know what p is, just put it in the calculator, 0.7, hit the x square button. 0.7 squared is 0.49. So it's asking us for a percent, so that's 49%. And heterozygous tasters, remember, heterozygous individuals, that's 2 times P times Q. I'm sorry, y'all, this is animating, so irritating. Here we go. 2 times P times Q, you just punch in 2 times 0.7 times 0.3, it comes up 0.42, so 42% are heterozygous tasters. Okay, so just remember, in any Hardy-Weinberg problem, and we're going to look at quite a few more, they're always going to give you some kind of number in the problem, a percent usually. And the key to solving these is you have to figure out what is that number that they're giving me. And here's kind of the rule of thumb. They either have to give you P or Q, or they have to give you Q squared, or they have to give you enough information to where you can figure out what Q squared is. And I'll tell you another little rule of thumb to maybe jot down here. It is always safest to solve for Q first. Figure out what Q is, and then base everything off of Q. Because if you know what Q is, then you can subtract from 1 and figure out the value of P. So don't ever, unless they just flat out give you the value of P, don't ever try to solve for P. Always find the value of Q first and base everything else, every calculation, off of that Q value. Most of the time, they're either going to give you Q as given information in the problem, or they're going to tell you what Q squared is, or they're going to give you enough information that you can figure out Q or Q squared. Just a quick refresher, P squared is the frequency of homozygous dominant individuals. 2 times P times Q is the frequency of heterozygous, so like big R, little r. And then Q squared is the frequency of homozygous recessive. All right, with that in mind, in a population of pea plants, 84% of those plants show the dominant trait of tall. Now, before we go any further, let me back up and tell you one more thing about this equation. If the given information in the problem talks about an allele, like 16% of the alleles are recessive alleles, or 84% of the alleles are dominant alleles, then they are giving you an allele frequency. They're either giving you P or Q. However, if they're talking about what percent of the plants, or what percent of the guinea pigs, what percent of the organisms, then they're not giving you an allele frequency, they're giving you one of these values. So in this problem, it says 84% show the dominant trait. So 84% of these pea plants are tall pea plants. Now think about it. What percent of the plants are short? If 84% are tall, that means 16% are short, right? They don't tell us that, but that's got to be true. So this might be a good time to pause the video, see if you can answer the 3 questions, and then unpause it and let's see how you did. Okay, now let's see how you did. I will tell you from the get-go, this is one of the trickier Hardy-Weinberg problems, and it's only the second one you've ever tried to solve, so it's probably mean of me to give you a tricky one. But the key to this is to figure out, what do I do with that 84%? 84% show the dominant trait. So think about this. Who would show the dominant trait? The homozygous dominant plants would show the dominant trait. But are they the only ones? No, the heterozygous, right? In other words, the big T plants, they would be tall, but so are the big T, little t plants. So the only plants that are not showing the dominant trait are the homozygous recessives. So if they're telling you that 84% show the dominant trait, that means 16% are showing the recessive trait, and the only ones that show the recessive trait are the homozygous recessives. So what does that mean? It means ignore the 84%, because we always want to try to solve for Q first. Remember what I said? It's safer to solve for Q, then you can worry about P. So if 84% show the dominant trait, that means 16% show the recessive trait. That's Q squared. So if I want to find P and Q, I'm just going to take the square root of 0.16, which is 0.4. 1 minus that is 0.6. So there's my values for P and Q. Now, what percent of the population is homozygous tall? I'm going to base everything I do now off of these two numbers. So what percent are homozygous tall? So that's 0.6 squared, which is 0.36, which is 36%. What percent are heterozygous tall? Remember, heterozygous is 2 times P times Q. 0.6 times 0.4 times 2 is 0.48. And you always have to pay attention. Are they asking for a frequency? Sometimes they are. Or are they asking for a percent? 0.48 would be the frequency, but they're asking for a percent, so I'm going to write my final answer as 48%. Okay, always good to remember, P squared is the frequency of homozygous dominant individuals. 2 times P times Q is the frequency of heterozygous individuals. Q squared is the frequency of homozygous recessive individuals. But if they're talking about genes, if they're talking about alleles, that's not going to be anything squared. That's just going to be a flat P or Q. So you have to sort of think about how the question is worded. Remember, you always want to solve for Q first, and then use this little equation to figure out the value of P. The given information in the problem pretty much has to either be Q squared or Q, or they're going to have to give you some information that you can use to quickly figure out the value of Q or Q squared. Let's try another one. And you can pause the video, work it out on paper, and then check your answer. It says the allele frequency for big T. So that's frequency of the dominant allele is 0.9. Pause it, answer the questions, and then unpause. So the key to this one is to realize that what they gave me in the problem was P. They gave me the flat-out frequency of the dominant allele is 0.9. That's given in the problem. 1 minus that is the value of Q, and that's what this question is wanting to know. What is the frequency of the recessive allele? So that's Q. And then what is the frequency of heterozygous tasters in the population? Heterozygous people who can taste is 2 times P times Q. So, and this time they're asking for a frequency, not a percent. So I could say 18%, but they're asking for a frequency. So you should always give them what they ask for, 0.18. So with a calculator, this is super easy. 2PQ. P is 0.9, Q is 0.1. Multiply them together times 2, and you get 0.18. Okay, always on all of these problems, I want you to pause it, Work it out. The only way you're going to get good at this is to practice. I promise this is one of the easiest things we do in this class if you'll just practice. But don't give up. If you're frustrated right now, don't give up, because after you do about 10 of them, you'll have it mastered, I promise. If 4% of a deer population are albino, which is a recessive trait, can we answer all of these questions? Now, always, we have to start, remember, by finding Q. Our goal is to find Q. On any Hardy-Weinberg problem, if you can find Q, you're home free. Because 1 minus Q is going to give you the value of P. And then you just plug everything else in. P squared, 2PQ, Q squared. So they always give you a number in the problem. What does that number mean? This is not telling me an allele frequency. It doesn't say 4% of the alleles are recessive alleles. It doesn't say that. It says 4% of the deer. These are living organisms that have 2 genes. 4% of them are homozygous recessive. They're little a because they're albino deer. So in other words, they're giving me Q squared. They are telling me that Q squared is 0.04. Now be careful, it's not 0.4. It's not 0.4, that'd be 40%. It's 4%, so that's 0.04. Okay, if I want to know what Q is, just take the square root of that. Square root of 0.04 is 0.2. Okay, 1 minus that is 0.8. Now I'm home free. Frequency of the big A allele, I already calculated that, it's 0.8. Frequency of the recessive, we already calculated that. Right there, it's 0.2. What percent of the population is normal, not albino? Now, there's 2 ways to solve that. Okay, the mathematical way is, let's see, normal deer. That would be the homozygous dominant deer, big A. That would also be the heterozygous deer, the ones that are big A, little a. So P squared plus 2PQ is 0.96, 96%. But you want to know what? The easy way? It tells me right here 4% of them are abnormal. 4% of them are albino. So the rest of them are normal, which is 96%. So I really didn't have to do any math on this one. It's pretty obvious that it's 96%. What percent of the population is heterozygous? That's always going to be 2 times P times Q, but they do want it as a percent. So instead of 0.32, we're going to call that 32%. Are you getting the hang of it yet? My gut says that you guys are probably getting the hang of this by now, but you know what? Practice makes perfect, so let's keep going. Okay, example number 5. 20% of the alleles are big A, dominant alleles. So we got to calculate all these frequencies. So what did they give me in the problem? They gave me a straight up allele frequency. 20% of the alleles, not the deer, not the plants, not the individuals, but the alleles are dominant alleles. So they gave me P. And 20% is 0.2. So that's the value of P. That's the frequency of the dominant allele. 1 minus that is 0.8, which would be the frequency of the recessive allele. Everything else is based off of those 2 numbers. So 0.2 squared is 0.04. 2 times P times Q is 0.32. And Q squared, let's see, 0.8 squared is 0.64. And so what is the frequency? Oh, this is an interesting question. What is the frequency of the dominant trait? You got to think about which individuals. We don't know what these are. We don't know if these are deer or flowers or what. It doesn't tell us. But who in a population shows the dominant trait? The homozygous dominant individuals, they show the dominant trait, but they're not the only ones. The heterozygous ones also show the dominant trait. The only ones that don't show the dominant trait are the homozygous recessives. And we know there's 64% of them, so that means the other 36% would have to show the dominant trait. 64% show the recessive trait, 36% show the dominant trait. And so the way I calculated that was p squared plus 2pq, but I could also just do 1 minus 0.64, and you can see that would give me the same answer. So I hope you're, on every problem, I hope you're pausing it, giving it a try. Even if you've done 5 of them incorrect in a row, don't lose hope. You'll get better at it. It just takes practice. Let's go to the next one. Okay, number 6. We know that big T is for being able to taste PTC. Little t is for non-taster. It says, totally made up here, it says 51% of a human population is able to taste PTC. And now we're going to calculate P and Q and everything else. So pause it. All right, and now let's see if you did it correctly. What are they giving me? They're telling me the percent that shows the dominant trait. Well, that doesn't really help me because I need to find Q. My goal is to always find Q first. So if 51% show the dominant trait, that means 49% of the humans, the rest of the humans in this population, must be homozygous recessive, right? Because those would be the ones that show the recessive trait, the non-tasters. They would be the little t people. And I know there's 49% of them because it says 51% can taste. That means 49% cannot taste. So I'm going to start with that. That's my Q squared. I always want to find Q first, so take the square root of 0.49. It gives me 0.7, and my P is just based off of that. Everything else is easy. Frequency of heterozygous people is 2 times P times Q. Frequency of homozygous tasters is 0.3 squared. 0.3 is P, so P squared is 0.3 squared, and it's not 0.9. You might think in your head, if you try to do that in your head, you might think, okay, 0.3 squared, that's probably 0.9. But I promise you it's not. Put it in your calculator. I'm actually putting it in my calculator right now. 0.3, and then I'm going to hit the X square button, and it is 0.09. Okay, you notice on these problems, whenever I'm calculating the genotype frequencies, like we did right here and right here, normally 2 decimal places is totally adequate. And I don't recommend ever going beyond 2 decimal places, except for a very rare occasion when you get a genotype frequency that's really, really, really close to 1, like real close to 1, like 0.99999, something like that. Or you might get one that's really, 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 really close to 0, like 0.00001. And that's pretty rare, but I'm going to show you one like that just in case you were to ever see it. You probably would never see one of these on the AP exam, but just in case, let's try it. So it says 99% of the human population has not inherited Huntington's disease, and that's a good thing because remember, it's a horrible neurodegenerative disease. And if you find out that you've inherited 1 copy of this dominant allele, you're going to die. I mean, it's inevitably fatal. So thankfully, at least 99% of the population has not inherited this disease. So we'll go off of that number. So that means, and so remember, it's a dominant allele that causes this disease. So 99% would be the ones who have a big H for Huntington's. That means 1% are little h. No, I'm sorry. I'm thinking of this backwards. Forgive me, and there's no way I can edit out my mistake, so let me start over. It says 99% has not, I didn't see my big word here, not inherited Huntington's. So those are the little h's. Okay, don't let me lead you astray here. 99% are little h. Okay. That means 1%, according to this problem, and it's really not it's really not as high as 1%, thank God. But let's assume that 1% has Huntington's. So theoretically, they could be big H, or big H, little h, although I think this one, homozygous dominant, would be probably fatal at the embryo stage. So really, people with Huntington's are heterozygous. That's neither here nor there. If 99% do not have Huntington's, little h, that means the other 1% theoretically are this or this. Okay, so that means that Q squared is 0.99. 99% are homozygous recessive, little h. Okay, so if I take the square root of that, I'll find Q. And you see I did more than 2 decimal places because otherwise it was just going to be 0.99 and then it would round up to 1. And I needed it to not be 1. I needed to be more precise than that. So you can see I carried it out 3 decimal places. 1 minus that gives me a p-value of 0.005. So this is a really weird problem. And it gets even weirder, because if you wanted to calculate the probability or the frequency of homozygous dominant, you take that 0.005 and square it, and you get this ridiculous number, 0.000025. If you wanted to know the frequency of heterozygous, that's 2 times p times q. So multiply these two numbers times 2, you get this number. And then we already know 99%. That's a mistake. Pardon me, I'll fix the mistake. So I just corrected it. So yeah, the Q squared is the homozygous recessive, and I promise I had little h, but sometimes autocorrect automatically wants to capitalize that because, it's autocorrect. It thinks it's a god. Anyway, sorry about that. I don't want to confuse anybody. But add these together. It's always a good idea at the end, if you add these 3 numbers together, p squared plus 2pq plus q squared, it should add up to 1. And if it adds up to 1, you probably did the math correctly. And you guys probably just checked my math, and I did the same thing, and it's technically not 1, but it's 0.999975, and that's pretty darn close to 1. So that's just from where we did some rounding. Okay, boy, let's move on to a less insane problem. Okay, so number 8, this is more of a typical problem. It says, seems like we had one like this already. 20% of the alleles in the gene pool are recessive non-taster alleles. So 20% of the alleles are little t. All right, so they are giving us what? They are giving us the allele frequency for the recessive allele. So they're giving us Q. Now 20% is 0.2, not 0.02. That would be 2%. This is 20%, so it's 0.2. 1 minus that is 0.8. Everything else is based off of that. And hopefully you're starting to get the flow here. All right. Ooh, another little wrinkle here. This was a little different. It says in a group of 100 people, so this is not alleles, this is organisms, individuals, people. 16 of those 100 people are homozygous tasters, 48 of them are heterozygous tasters, and 36 of these people are non-tasters. Those are the ones that we really kind of want to focus on, because remember, it's the homozygous recessive people that show the recessive trait. So that means 36 people divided by 100, that's 36%, are homozygous recessive. So what did they give us in the problem? Well, based off of that 36 divided by 100, that's 0.36. So that would be our Q squared. You follow me so far? If you got this far, I know you can do the rest, because you just take the square root of that. Square root of 0.36 is 0.6. 1 minus that is 0.4. Okay. And that's the last Hardy-Weinberg problem. Now, I've got a lot more Hardy-Weinberg stuff to talk about, as you see here, but I'm going to save that for another lesson, because after all that math, your brain's probably smoking and your calculator battery's probably dead. So we'll stop right here, and I hope you enjoyed it, and I hope you have a great day, and I'll see you next time.